In this lecture, we are going to take a look at some pathophysiologies that impair or restrict our ability to ventilate. So the first thing to understand is that the lungs must be stretchy in order for ventilation to occur. So when you push down on your diaphragm and you expand out your rib cage, we expect that the lung tissue will go with it. So during inspiration, the lungs are stretched, and during expiration, the lungs recoil. So we have a term that we apply to describe how well the lungs stretch out and how well the lungs recoil. They are compliance and elastance, respectively. So we're going to start by taking a look at compliance. We'll formally define it, and then we'll talk about some pathophysiologies that impair compliance, and then we'll do the same for elastance. So compliance is the ability of the lungs to stretch. So when you look at the alveoli that are attached to the inner pleural membrane, the alveoli are comprised largely of simple squamous epithelial cells. So that's just a very thin layer of cells, much like a thin layer of fabric. But there's also elastin, which is a type of elastic fiber that's woven in between. So as a consequence, it kind of behaves like spandex. So when you pull your rib cage out and your diaphragm down, we expect that the alveoli are going to stretch like so. And a good stretch in that particular lung tissue is a compliant lung tissue. So we define stretchy lungs, ones that stretch easily, as being very compliant. If there is a disease that reduces compliance, it means that it's really difficult to extend or stretch the lungs. In this way, the lungs are going to act as if they're more wooden and not as compliant. And it would absolutely impact your ability to inspire or bring air in. So what could make our lungs less compliant? Well, one thing that will definitely make our lungs less compliant is if the lungs have scar tissue in them. So that is because then the scar tissue makes for thick lungs or thick alveoli. Imagine if I take this, what was a compliant piece of fabric, and I fold it over 4 different ways, so now that it's 4 layers thick, and now I try and stretch that. It's much more difficult to stretch. I mean, I can still do it, but I have to pull a lot harder to get it to stretch. So in the same way, when you take your lung tissue and you add scar tissue to it, scar tissue is not stretchy and it's thick. It makes it a lot more difficult for your lungs to comply. So what kind of diseases might induce the buildup of scar tissue? Exposure to any sort of irritant that causes damage to the lungs, or a toxin that causes damage to the lung, or even certain types of infectious diseases can cause permanent scarring of the lungs. This can result in fibrosis. So fibrosis is usually a name that we apply to the development of scar tissue. And you can see here that on the left, the lung tissue looks very thin, and on the right, it's much thicker. One potential notorious cause of fibrosis is asbestos, which is shown here. Asbestos was used historically as an insulation material in homes. The problem is that asbestos also has these tiny little, almost glass-like spicules that very easily get dislodged and aerosolized. So when people would install asbestos or handle asbestos, these little pieces would fly into the air and get inhaled. And when they would get inhaled, they would actually land in the lungs themselves and cause scarring. So scar tissue, like fibrosis, which can be caused by a number of things, including asbestos, reduces compliance. Another way that compliance can be reduced is by lack of surfactant. So recall that type 2 alveolar cells secrete surfactant, and surfactant makes it so that the hydrogen bonds between water molecules are lessened, and therefore makes it so that the alveoli walls are not as likely to stick together. This is actually described in more detail in Lab 18, where I have 2 microscope slides and I put some pure water in between them and then stick those microscope slides together, and they do stick. And then I ask my sons to try to peel them apart, and it's really difficult to do because the water molecules are hydrogen bonding with each other and hydrogen bonding with glass. But if you throw in something that has a surfactant-like quality to it, soap, contact lens solution, and then you try to stick those 2 microscope slides together, they peel apart much easier. So surfactant is super important in ensuring that the alveoli do not stick together kind of permanently and that they peel apart really easily again when you try to pull them apart during inspiration. So if anything impacts the type 2 surfactant secreting cells, you can end up with reduced compliance. And one example of this is going to be neonates. So remember, in utero, a fetus is not breathing. They have no need to breathe because they're getting all of their oxygen from mom. But upon birth, that connection to mom is severed. And so now the newborn has to breathe to live. And when it breathes, it needs to pull in fresh oxygen, and that's going to be the way it's going to oxygenate its blood. Well, what happens is that in some cases, especially with premature babies, the type 2 cells are not mature, and so they don't secrete enough surfactant. And this can lead to respiratory distress syndrome, where the alveoli stick to each other upon exhalation or expiration, and then the baby can't get them back open again. And so there's a number of interventions for this, but without medical treatment, it can be fatal. The baby will be chronically cyanotic, blue, because they're not able to get enough oxygen. They've got kind of a grunting respiratory response that's abnormal. And again, it's much more common in premature babies. It's particularly those born under 30 weeks of age. And then also common in cesarean sections. And the link to cesarean section probably has to do with the fact that there's a number of physiological changes that the baby themselves undergoes as part of the birthing process, meaning that, they themselves are getting feedback from mom, like, you're going to be born, get ready. In a cesarean, some of those feedback mechanisms are missing because they don't pass through the vaginal canal with, a prolonged labor. There's just a cut to the uterus and they're taken right out. So that probably has something to do with it. In modern times, there's a number of interventions that certainly help mitigate respiratory distress syndrome, but without that care, it can be a cause of fatality. Now, respiratory distress syndrome certainly can be caused by immature surfactant secreting cells with neonates, but it's not the only cause of a respiratory distress syndrome. Any sort of damage to the surfactant secreting cells can also cause respiratory distress. And one cause of that is COVID-19. So COVID-19 actually gets in and infects your surfactant secreting cells. Well, remember that when you have an infected cell, your cell-mediated immune response is to kill that infected cell. And so this can lead to respiratory distress. And it's actually one of multiple mechanisms leading to respiratory distress when your immune system kills off your own infected cells. And those cells were necessary for preventing alveolar collapse where you couldn't open them back up. But as you can see from this picture here, it's a little more involved than just that. And it makes an interesting case study because it combines both immunology and respiratory physiology. So we're actually going to put a pin in this and take a look at COVID-19 a little bit later on in this lecture series. So you'll see this one in a couple more lectures. Now let's turn and take a look at elastance. So elastance is the ability of the lungs to recoil. And this is largely due to the elastic fibers, which are elastin. If you have a reduction in elastin, then your lungs are going to be like stretched out gym shorts. That means that they might be stretchy, but when you let them go, they don't recoil. So Play-Doh is a really good example of something that has great compliance but no elastance. You can stretch it and then you let go and it just sits there like, I'm not recoiling. So in the same way, if you don't have good elastance, that can make it very difficult to exhale air. And people that have reduced elastance often look like they're kind of wheezing or puffing, like they're forcefully trying to exhale air, which tends to be more of a passive process. When we think about, is it more difficult to inhale air or exhale air? Most people think it takes more work to inhale air, and then the exhalation is kind of a natural process. It's just very passive. And again, that's largely because our elastin, elastic fibers, are doing some of that work for us. If they are damaged, they're not doing that work for us, and we have to do that work by forcefully compressing our rib cage, which takes a lot more work, and getting that air out. So one of the causes of that is emphysema. So emphysema is a disease that has a number of triggers, but can be related to asthma, it can be related to chronic smoking or chronic irritant exposure. And what happens is we end up with damaged elastin and actually damaged alveolar walls. So it's going to create a myriad of problems for us. So let's go ahead and spell this out. So emphysema is, again, characterized by loss of elastin. The elastin is usually damaged. And as a result, we have lack of elastance. It's very difficult for people with emphysema to expel the air, and so they look like they're working or puffing to kind of push out that air. Emphysema is a common cause of something called chronic obstructive pulmonary disease. And obstructive pulmonary disease usually means that we're having difficulty getting the air out. The fact that emphysema also damages the alveolar walls, and hopefully you can see that here compared to here, also means that there's less surface area in the alveoli, and we'll address the implications of that in another lecture. I also want to point out that chronic emphysema can also lead to buildup of scar tissue, depending on the cause, and therefore we can see a reduction of compliance as well. And so while emphysema is characterized by a loss of elastance and a reduction of surface area that's going to impair gas exchange, as we'll see in another lecture, you can also have a reduction in compliance associated with this disease as well. So not all ventilation problems are going to be restricted to the alveoli. You can have obstructions in the airway too, in the bronchi and the bronchioles. And if you have an obstruction in the airway, that can also make it very difficult to ventilate. So I want to give you an analogy. Imagine that you are going to affix a straw to your lips. So you're going to tape a straw to your lips and you're going to plug your nose and you have to breathe through that straw. These are your choices. Which straw are you going to pick? Well, I don't know about you, but I would probably pick this one right here, the biggest one, and I'd probably want it cut to make it as short as possible. I certainly wouldn't want one of these long, skinny ones. In the same way, when we look at our lungs, having big open airways makes it much easier to move air because there's less resistance. Now, if I were to try to make these all like the skinny, tiny coffee straws, it would be a lot harder to move air through those tiny diameter airways. So we saw this also with like the cardiovascular system and resistance, but it also plays a role in the respiratory system. The larger the diameter of your airways, the easier it is to move air from here to here because there's less resistance. So what are some things that can obstruct the airways? So one of the ways that we can obstruct the airways is to actually clamp down on the smooth muscle that surrounds the airways, particularly the bronchioles. Remember that bronchioles have nothing but smooth muscle around them. They have no cartilage plate reinforcement. So if you take that smooth muscle and you constrict it, you could completely pinch off the bronchioles. So remember that smooth muscle is either going to be controlled by the autonomic nervous system or by local paracrines. So the bronchioles are largely controlled by paracrines. And so let's take a look at some things that might impact the smooth muscle around the bronchioles and their impact on diameter. So briefly, before we look at this, imagine if I have an airway that looks like this. That's a nice open airway, good flow of air through there, right? No issues. But that also means that the smooth muscle that's around that is nice and relaxed. So this is going to be relaxed smooth muscle. Conversely, if I tighten that smooth muscle, I can constrict that airway substantially. So here I'm going to really tighten it up. And now it's much more difficult for air to get through. So by changing the tone of the smooth muscle around the bronchioles, I can change how easy or difficult it is to move air between the atmosphere and my lungs. So histamine is a super important paracrine that affects the smooth muscle. And histamine causes bronchoconstriction. Bronchoconstriction is the name that we give to a bronchiole or a bronchi that has been constricted and its airway reduced. So in fact, let's go ahead and label these as bronchodilation. That's where the smooth muscle is relaxed. And bronchoconstriction can result when the smooth muscle is constricted. Okay, so histamine is a powerful bronchoconstrictor. Now, histamine is released as part of the inflammatory response. Here, the culprit probably wouldn't be basophils as much as it would be mast cells that are stationed in the area. I'm going to come back to histamine when I talk about asthma in just a moment, but let's take a look at some of these others. Epinephrine and norepinephrine can also act on the smooth muscle. In which way? Do they cause bronchoconstriction or bronchodilation? Well, what would make sense? Epinephrine and norepinephrine are part of the sympathetic nervous system. Imagine if you need to think clearly and run away from a dangerous animal that's chasing you. Would it make sense to get more air into your lungs so that you get more oxygen for sustained aerobic activity? Or would it make sense to shut your lungs off and so that you fall over and get eaten by the animal? Hopefully it makes sense that you would want your lungs open so that you get as much oxygen as possible so that you can sustain running away from the animal or the danger or whatever it is that you need to deal with. So epinephrine is a bronchodilator. It helps open up your lungs. Now CO2 is an interesting one. CO2 is produced as a byproduct of aerobic respiration. And so we have alveoli, and I'll go ahead and draw kind of a classic alveolus there. And we have blood that's coming by. And we have CO2. I'm going to ignore the oxygen here. And CO2 is getting dumped into the alveoli. So CO2 starts accumulating in this alveoli. Now I need to get that CO2 out. For reasons that are not clear yet, but hopefully will be by the time we are done with this particular lecture series, The CO2 amount in the blood is very tightly regulated, and therefore the CO2 in the alveoli cannot build up or you're going to be in trouble. So when CO2 accumulates in the alveoli, it has a paracrine effect on smooth muscle rings that control the exit or escape route for that alveolus. If there's too much CO2, my number one priority is to get that CO2 out. The way that I get that CO2 out is by opening up this bronchiole here and making it wider so that it's easier for CO2 to exit. The way that I do that is by relaxing the smooth muscle. So when CO2 levels are elevated in the alveoli, it actually causes upstream bronchodilation. And upstream means that we're looking upwards from that alveolus, what bronchioles are serving that alveolus, and we need to make sure that the airway is nice and open to get that CO2 out. The last thing on my list is albuterol, and albuterol is used in asthma inhalers. So just reasoning it out, I bet you could guess what albuterol probably does. It counteracts the effects of asthma, and as we will see shortly, the effects of asthma are to cause bronchoconstriction. So albuterol is a powerful bronchodilator. Okay, now that we've introduced some paracrines that will help modulate the smooth muscle tone around bronchioles and therefore the ability of air to move through the airways, let's take a look at asthma. Asthma is perhaps the most common disease of the airways in this way, and it affects a significant percentage of the population. Asthma is a disease where mast cells oversecrete histamine. There can be a number of triggers to this, but the outcome is more or less the same regardless of the trigger. By oversecreting histamine, that is going to cause bronchoconstriction, making it more difficult for air to get into the alveoli. But moreover, histamine is also a major contributor or instigator of the inflammatory response. And do you remember what one of the things that occurs in the inflammatory response is that blood vessels nearby become leaky, such that fluid can move out of the blood vessels and into the interstitial fluid. Except here, the fluid moves into the airway. And so we end up with additional fluid accumulation that further restricts the useful airway. So you can see that here, hopefully. You can see the bronchodilation, and then you can see the fluid accumulation that further takes up very valuable airspace and restricts that airspace even farther. So asthma is a disease or a disorder where mast cells are prompted to oversecrete histamine, and as a consequence, we have a double whammy. So that double whammy is bronchoconstriction, and inflammation, which causes fluid accumulation in the airway. All right, and with that, we'll end this lecture. And in the next lecture, we are going to start looking at gas exchange between the alveoli and the blood.